Hereditary Non-Polyposis Colorectal Cancer by Jalen Calloway
and Michaela Ivy. The purpose of our experiment was to do functional analysis of MSH2 mutations. This cannot be done in humans. Inserting a mutation in humans is unethical and could lead to legal trouble. Analysis of MSH2 gene in yeast will give us similar response because the gene sequence and functions are very similar.
MSH2 plays a vital role in the mismatch repair system. This system repairs DNA damages like insertions and deletions by means of incision. Our restriction enzyme was BSMA1, cutting the mutation G704D. This mutation is cloned from the E. coli strain.
Hereditary non-polyposis colorectal cancer takes place in the intestines. Hnpcc is a genetic disease of autosomal dominant hereditary cancer.
It is caused by a mutation in the MSH2 gene of the DNA mismatch repair system. If there are multiple mutations present that have not been corrected by MSH2, the risk for developing Hnpcc increases.
There is not a visible formation of polyps along the colon. Patients do not experience any physical symptoms. Major signs of Hnpcc is blood present in the stool, diarrhea, chronic constipation, decrease in amount of stool, gas, bloating, vomiting, or weight loss.
A change in single nucleotide can affect the function of the MSH2 gene. A single nucleotide change can cause an amino acid to be changed, causing a missense mutation within the sample. In the mutant, there is a missense mutation present, found by using APE to compare the DNA sequencing and using Clustal to compare the amino acids.
For our experimental design, we harvested yeast cells to use as the DNA, isolated the plasmid DNA from E. coli strands of bacteria, which generated concentrations of the samples. We digested the enzymes using XBAI and APA1 to generate the identities of each strand, and we used PCR to amplify the MSH2 gene while using a restriction enzyme generated by NEB cutter.
The isolation of the plasmids, the E. coli 103.6, 455.9, and 300.4. The ratio, the 260 to 280 ratio, defined the purity of DNA. It was the absorbance of proteins over nucleic acids. A value of 1.8 generally signifies pure DNA. If the ratio is lower, that would signify contamination of DNA plasmids. Our ratios depicted 1.87, 1.74, and 1.73.
And here are predicted outcomes of the Argos gel electrophoresis using APE. APE showed the predicted fragment sizes as well as specific locations in which the restriction enzymes will cut the plasmids.
The RE digestion lab, it identifies the plasmids. SPO cut had 7.0 kb and 3.0 kb. SPO uncut had 5.0 kb. SP1 cut had 10 kb and 6 kb. SP1 uncut had 9 kb. SP3 cut had 8 kb and 3 kb, and SP3 uncut had 9 kb and 5 kb. These bands show the places where XBA1 and APA1 cut the DNA sample.
Next, we did a PCR reaction, which copies the MSH2 gene. First, we denatured, which takes place around 92 to 95 degrees Celsius. This step breaks the bonds and causes the double-stranded DNA to become single-stranded. This gives polymerase the opportunity to attach. Next was the annealing step. The primers are given a chance to form base pairing with the template strand in 50 to 60 degrees Celsius. Next is the extension stage, which adds nucleotides at 72 degrees Celsius. And next, PCR was used to amplify where the MSH2 gene is located on the DNA sample and where the restriction enzyme cuts it.
The mutation in SP3, these two photos are of the two-strand alignment through RStudio and Clusto with colors, and it gives you the position on both of them, and the position on Clusto was around 720 between that on the mutant and the wild type, and then the other one was found through the two-strand alignment on the line that reads 5,500.
Here is our restriction enzymes for the mutant. Our restriction enzyme was BSMA1, and we chose that only because it cut our mutant strand.
Okay, so next we did the gel electrophoresis of PCR products, and it showed us the base pairs. So the SP3 cut had an 1,000 base pair, an 800 base pair, and a 100 base pair. The SP3 uncut had a 1,000 base pair, an 800 base pair, and an 100 base pair. And the SPO was uncut over uncut. And it says the SP1 cut had an 1,000 base pair, and the SP1 uncut had an 1,000 base pair as well.
And during this lab, we had an error. Some of our restriction enzyme likely got into another well, causing cuts in unnecessary places. Next part of our lab was our yeast transformation. We plated the mutant and plasmid MSH2 to measure the mutation rate of each. We did this by denaturing the carrier DNA with 1,000 degrees Celsius heat block and chilled it. And we prepared a positive and negative transformation tube. Inside the negative tube, the DNA was replaced with water, and then we heat shocked the cells at 42 degrees Celsius. And we centrifused and plated both cells on a media that did not have His, TRP, and URA.
So here are our yeast transformation results. We had an error on the plate with the DNA. There was a growth of fungus, likely because there was contamination from breathing or exposing the sample to too much air when we plated in the dish.
And here's our FOA assay plate. We did this experiment by diluting the samples of SPO, SP1, and SP3. Okay, then we plated them onto a plate of SD medium containing FOA and all the essential nutrients except histidine, tryptophan, and leucine, and threonine. The empty vector produced the most mutations. The mutant strand, our SP3, produced less mutations than that of the empty vector in MSH2. The mutation in MSH2 is needed to combat the mutation rate of a cell. It decreases the abundance of mutations.
So for the flow symmetry, the HU treated and the untreated cells. So flow symmetry means the measurement of cells. Any cell that can be suspended in a liquid like bacteria, blood cells, or a plant cell are capable of being placed in a flow cytometer. The flow cytometer is an instrument that can evaluate the single cells. It does this by allowing the cells to flow through a chamber and pass by a laser that collects signals given by the cells and then are collected by a computer. The data collected reveals the cell's shape, size, and other special properties.
We began doing our flow symmetry by first arresting DNA replication by using hydroxyurea in one of our samples. The HU arrests the cell cycle and puts every cell at the G1 phase. We did this to one tube of our mutant while the other was HU negative. The HU plus cells were washed and media was added back to it to allow the cells to grow. Next, cell division was reactivated and the damaged cells were fixed by ethanol because it fixes the cell structure.
So the HU treated cells were mostly round and are arrested at the G1 phase, as said before. This is because hydroxyurea induces cell arrest. Approximately 18.5 of the cells of the HU plus are shown budding, and approximately 52.3% of the cells in the HU minus are shown budding.
In conclusion, our SP3 was our mutant, our SP1 was our wild type, and SPO was our empty vector. To cut SP3, we used BSMA1. And during the first restriction enzyme lab, the plasmid was cut by XBA1 and APA1 because they contained MSH2. The SPO in the gel electrophoresis had smaller fragments because it did not contain MSH2 genes. This notified us that that was our empty vector. Some errors may have come from contamination because we forgot to change the tips, and miscalculations due to dilutions. Also, our SNP mutations can combat the formation of mutations that lead to Hnpcc. These mutations are needed to decrease the frequency of polyp formation.
Thank you so much for watching our presentation. Here are our references.